I am Professor Amer Jamil from Department of Biochemistry, University of Agriculture Faisalabad. This lecture is part of the series of lectures on basic molecular biology. So in this lecture we will be discussing about translation. Translation is synthesis of proteins both in prokaryotes and eukaryotic cells. Translation involves all three types of RNA molecules. Messenger RNA carries the message and transfer RNA carries the corresponding amino acid, whereas ribosomal RNA are part of the ribosomes where this translation takes place. Translation takes place on free ribosomes for the proteins which have to be remain inside the cell, and the translation takes place on ribosomes attached to the endoplasmic reticulum for the proteins which are to be exported either out of the cell or inside the cell membrane, or maybe in eukaryotic cells in another part of the cell. Translation takes place using a certain reading frame which is a sequence in the messenger RNA, and this sequence is composed of codons. Each codon contains 3 nucleotides, and each codon codes for one amino acid. So there is a probability that which kind of reading frame can be used. Like here there are 3 reading frames. It depends from where this translation begins. If it begins from this U, these will be the codons. For reading frame 2, it may be started from this second nucleotide, and you can see that there is total change in the codon sequence which will make it entirely different protein. And for the reading frame 3, it will be started from the third nucleotide, and once again this shows entirely different sequence that will correspond to different protein. It means this reading frame is very important for translation. There are total number of 64 codons. Out of these 64, 61 code for 20 amino acids. So as you can see here, these UUU for phenylalanine, and similarly you can see many amino acids have more than one codons, because codon number is 61 and amino acid number is standard amino acids are 20 amino acids which are protein bound. And the 3 codons which do not correspond to any amino acid, they are called as stop codons. UAA, UAG and UGA. These are the 3 stop codons. And there were many scientists who actually devised procedures to reduce this genetic code. So this is the genetic code dictionary, and Codonsa is one of the major part of the team that actually deduced this genetic code. We will see there are some amino acids, actually 2 amino acids who have only one codon. So rest have more than one. And methionine has an important position in translation, because almost all protein translation starts with AUG as the initiating codon, and it codes for methionine. Here you can see that 2 amino acids, methionine and tryptophan, have one codon, whereas many have 2, isoleucine has 3, and some have 4 codons. And 3 amino acids, arginine, leucine and serine, they have 6 codons. This is called as degeneracy of the genetic code. It has, from where it comes, as I mentioned earlier, there are 61 codons for 20 protein bound amino acids. And total number of 64 codon, how those are possible? If you see total number of nucleotides, types of nucleotides are 4, and number of nucleotides in a certain codon is 3. So if 4 is to power 3, it means 4 into 4 into 4 comes out to be 64. So 64, there is a possibility of 64 combinations of these 3 nucleotides in the form of codons. So it means there are 64 codons, and out of these 3 are the stop codons. But there is, in rare cases, some variation also exists in the codon assignments. For example, here you can see the variation in codon assignments in mitochondria. Like I give you the example of UGA, which is usually a stop codon, but it codes for tryptophan in mitochondrial genome of these organisms. Similarly, AUA is for isoleucine in the normal codon assignment, for methionine for in the mitochondria of these organisms. But it has, in other microorganisms or other organisms, it has the same codon assignment as AUA, even in mitochondria. So the point here is that there is a variation that may exist in codon, in the genetic codon assignments, which are which come under the standard genetic codes. So there may be some variation. And this is a typical type of anticodon-codon interaction. This is with the help of hydrogen bonding, and both of these are complementary to each other. And this anticodon is present inside the transfer RNA, on one side of the transfer RNA, and codon is present in the messenger RNA. And by convention, this messenger RNA is written in the 5' to 3' direction. So the 5' direction of transfer RNA is on this end because of the anti-parallel nature of these two molecules, because they both run anti-parallel. However, this transfer RNA, as you can see, its structure is it has a lot of intra-chain hydrogen bonding, which makes this structure. I will explain in a short while what is the significance of this type of structure. And you can see how these codon and anticodon, they interact with each other with the help of hydrogen bonds. And you can also see inosine present in anticodons in many cases. 5' base present in the 5' end of the anticodon, and the base present at the 3' end of the codon. They are called as Wobble bases, and they are shaded in pink in this figure. The name was given by Crick, the same person who deduced three-dimensional structure of DNA along with Watson. So these X and Y represents the normal Watson and Crick base pairing as GC and AU. But these, here we will see there is some variation. So in some cases, only one codon is recognized by this anticodon, like C recognizes G, A recognizes U. But in other cases, there are two codons that are recognized by this anticodon. For example, if it has uracil in 5' position, it may recognize either A or G. Similarly, if it has G in the 5' position, it may recognize C or U. And if it has inosine in its 5' position, it may recognize any of these three, A, U, and C. So it means this base wobbles, but these two remain the same. So these two determine basically the which amino acid will be coded by which codon, but the third nucleotide, it gives degeneracy to the genetic code. So certain frame is very important, as I mentioned earlier. So if there is some frame shifting, as in case of retroviral transcripts, these are the viruses, RNA viruses. So you can see in one of the gene, GAG gene, it is coded by this messenger RNA, and this is the stop codon. But another one, like Paul reading frame, this Paul is another gene that codes for another protein. So it starts from here. So you can see there is some sort of overlapping here, and then there is some sort of frame shifting, so that this is not the stop codon. RNA editing may also play its part, that what kind of protein is going to be made. So as in this case, this is the DNA coding strand, but you can see in messenger RNA there is some changes. There are some changes, like here it should have been C, but it is U. So it means there is editing of the RNA that takes place, which making this to U. Similarly for others as well. So basically there is maybe some deamination takes place, or as you can see, if addition of water will convert this adenosine to inosine, but on the other way, if there is a deamination takes place of inosine, it becomes adenosine. And similarly, it may oscillate between cytidine and uridine. So it means in case of RNA editing, you get different protein. Like here in this case, uh, RNA editing of transcript of the gene for ApoB100 complement of LDL. So what is this effect on deamination? As you can see in human liver, ApoB100 produces this protein, while in intestine you can see a shorter protein is made, because this part, this C, is converted into or edited into U. So if U is there, it becomes UAA, which is a stop codon. So you get a shorter protein in human intestine as compared to the human liver, where this is not the stop codon, it will go on. So you can see also the difference in their molecular mass as well. So it means there is a regulation at the translation level as well, where this editing in the messenger RNA makes a different protein in different parts of the tissues depending on the need of the cells. So if we look at the components of ribosomes, so here the components of E. coli ribosomes, they have two subunits, 30S and 50S, and number of different proteins which are present. There are many identical proteins, but there are different proteins, like in 21 proteins are present in 30th subunit and 33 in 50th subunits, and total number of proteins, meaning some may not be different, so total number is 21 and 36, and these proteins are designated as S1 to S21 and L1 to L36. Similarly, the ribosomal RNA which exists in 30S, it is 16S ribosomal RNA, and in 50S it is 5S and 23S ribosomal RNA. I discussed in detail about synthesis of this ribosomal RNA, both in prokaryotes and eukaryotes. You can check on that lecture if you want to. So the bacterial ribosome contains three important sites: E for the exit site, peptidyl site, and aminoacyl site. So these are the three important sites in bacterial ribosome. And this messenger RNA, you can see it is shown here. So there is a certain sort of pocket inside the ribosome where this messenger RNA comes and starts this translation process. So here there is a comparison of bacterial ribosomes with the eukaryotic ribosomes. So you can see difference in the molecular masses and all that, and this is 50s and 30s subunits, whereas this in this case it is 60s and 40s subunits. So this 40 and 60 should have been made to 100, but actually it is 80s. Similar is the case here. So this S is the Swedberg constant. It comes out from sedimentation coefficient by multiplying with some number, so that we get a meaningful and easy to manipulate number rather than a long number with many zeros or decimals. So you can see here these are different types of RNA molecules which are present in both cases, but there is some variation as you can see here. And similarly in this case you can see there is 16S ribosomal RNA and 18S ribosomal RNA in case of this eukaryotic ribosome. So we have this 18S ribosomal RNA in this case. This is a nucleotide sequence of yeast tRNA alanine. So this is the anticodon for alanine, and alanine will be attached to this 3' end of the transfer RNA. This is secondary structure of transfer RNA, and it is a cloverleaf structure. There are different arms of this transfer RNA. One is the amino acid arm, where there is a CCA is present at the 3' end. Then there is a T5C arm. Phi is an unusual amino acid. There is an extra arm that is variable in size, and this is the Anticodon arm, and this is the Wobble position. And then there is a D arm that contains 2 or 3 D residues at different positions. So this is there is a lot of intra-chain hydrogen bonding that gives this structure, and this is a sort of adapter molecule, because on one end it will be interacting with the messenger RNA, and on other end it will be making the protein chain. This is again three-dimensional structure of yeast tRNA phenylalanine, and it is deduced from X-ray diffraction analysis, and these are the similar types of arms. Now coming to the translation, there are 3 steps of translation as for replication and transcription: initiation, elongation, and termination. But before initiation there is activation of the amino acids, and after termination there is folding and post-translational processing that takes place. And these are essential components of each stage of translation. So we will discuss these one by one. Amino-acyl transfer RNA synthetase. This is a very important enzyme in translation because it transfers amino acid to the transfer RNA. So it has to be very specific, because the amino acid which is transferred to a certain transfer RNA should have corresponding anticodon present in the transfer RNA. So this amino-acyl transfer RNA synthetases, there are two classes, class 1, class 2. Class 1 transfers these amino acids and class 2 transfers these amino acids. So if a wrong amino acid is transferred to some anticodon, like for example, if anticodon contains the sequence for methionine and on the transfer RNA, alanine is transferred, so you can imagine what kind of protein it will make. This is overall picture of activation of amino acids, meaning attachment of amino acid to the transfer RNA. Here it is more clear, like first of all what happens is there is ATP is involved and AMP is attached to this amino acid at the carboxylic group. So there is an attack of oxygen at this phosphorus, and this pyrophosphate is released, and this adenosine monophosphate is added to this carboxyl group of the amino acid, and it becomes 5-aminoacyl adenylate. Now this is brought about by the same enzyme, amino-acyl transfer RNA synthetase. Now there are two classes of the enzyme, as I mentioned. So half of the amino acids, they go through this pathway, another half through this pathway. What is the difference between two pathways? It is very minor. There are two possibilities now where this amino acid can be transferred to the transfer RNA. So this is the transfer RNA, and at the 3' end is the CCA. So this is the final A adenine. So there is a 2' hydroxyl and a 3' hydroxyl where this amino acid can be transferred. So if it is transferred at this place, it is brought about by class 1 synthetase. But in other amino acids, it is transferred to this 3' hydroxyl. So this is the difference between two classes. So this difference, as I mentioned earlier, may be due to create specificity, because amino acid should be attached with reference to the anticodon is present. So those amino acids, they should be attached specifically to their corresponding transfer RNA. That may be the reason that it is divided into two different paths. So in the next step, now this is the situation like in class 1, it is transferred at this place, and in class 2 it is transferred at this place. But later on, the amino acids which are transferred to 2' hydroxyl, they are also shifted to the 3' hydroxyl by transesterification step. So ultimate result is that all the amino acids now they are present at the 3' hydroxyl of this adenine, and this is called as amino-acyl transfer RNA, and it means now this amino acid is activated and ready for initiation of translation. This is the structure, overall structure of the amino-acyl group attached to the adenine, and this is the transfer RNA. So there are certain nucleotide positions in transfer RNA that are recognized by amino-acyl transfer RNA synthetase to make it specific. So the blue dots are the same in all transfer RNA molecules, therefore they are not used for this discrimination. But the orange dots or the green dots, they are used for this purpose. for the specificity. So you can see there are certain green dots and then orange dots. So they bring about this specificity. This is the structural elements of tRNA alanine that are required by for recognition by alanine tRNA synthetase. So in this case, this is the shaded portion that is important. Now that the activation of amino acids is complete, so we have seen how it takes place. Now comes the initiation of translation. The first amino acid that is added to the protein or the polypeptide chain is almost always methionine, whether prokaryotic cells or eukaryotic cells. But there is one difference in the prokaryotic translation, that it is N-formyl methionine that initiates the translation, whereas in eukaryotes it is methionine that initiates this translation. So this is the formyl group present at the N position of this methionine. So this is the formation of initiation complex in bacteria, and here you can see what are the requirements for initiation that will come in the slides. So it will be more clear from this. Like at first there is a 30S ribosome which contains P and A site. Then there are initiating factors. And remember, this is a translation in case of prokaryotes. So initiating factor 1 and 3, they bind with this 30S subunit, and then messenger RNA along with its codons, it comes on this 30S, and this messenger RNA scans the 30S ribosomal subunit so that AUG, the first codon, the initiating codon, which codes for methionine, it comes at the P site. There is a certain special sequence that allows AUG to come on this P site that I will explain shortly. Then comes initiating factor 2 along with GTP. This translation is a expensive process. Making a peptide bond requires energy, quite a good amount of energy. So this GTP is attached, and this brings this first transfer RNA along with F-methionine, and this is the anticodon corresponding to the codon AUG. So this whole complex forms, meaning this initiating codon in the messenger RNA, and the transfer RNA contains this F-methionine along with the anticodon along with GTP. And now this GTP is hydrolyzed to GDP and phosphate, and all the initiating factors, they are released. Now comes 50S subunit. In the picture you can see this is 50S subunit, it has E site. This next codon is ready to accommodate the next transfer RNA along with the amino acid. So the transfer RNA with the anticodon corresponding to this codon will come in place. Before going further, this is the Shine-Delgarno sequence after the name of the scientist who discovered this, and these are different messenger RNA for different genes in bacteria, in E. coli. And this is the Shine-Delgarno sequence. This sequence helps placement of initiating codon at a proper place uh, on the ribosome, on 30th ribosome. So this messenger RNA scans this 30th ribosome subunit, and this AUG finally it comes on the P side. And what makes this Schindel-Garno sequence is attachment to the uh, ribosome is the sequence in the 16S ribosomal RNA. You might remember that this 16S ribosomal RNA is present in the 30th subunit of the ribosome in bacteria. So this 16S RNA has a complementary sequence to this Schindel-Garno sequence. Whereas in case of eukaryotes, this 5' cap and 3' poly A tail play this important role of placing this AUG at a proper place. So, and these regions are called as UTRs, untranslated region. Whether it is in prokaryotes or eukaryotes, the region which is not translated is called UTR, untranslated region. So on the 5' UTR, and at the end is the 5' cap that is held by this initiating factor. Here for the eukaryotes, this small e shows eukaryotic. IF4E holds this 5' cap, and similarly this poly A binding protein holds this poly A tail. So this is the role of 5' cap and 3' poly A tail in translation. These are the initiating factors along with their functions in case of bacteria, and these are the initiating factors along with their functions in case of eukaryotic cells. And you can see a lot more initiating factors are required in case of eukaryotic cells, having different functions like binding to messenger RNA, and then 5' cap attachment, poly A binding, and so on, helicase activity in one case. So there are multiple eukaryotic factors, initiating factors having different functions. This is the first elongation step in case of prokaryotes with the binding of the second amino cell transfer RNA, and this is brought about by TU factor along with GTP. So this GTP is hydrolyzed and this TU factor is released, and this transfer RNA along with the amino acid, it comes at this place based on this anticodon, which is complementary to this codon, and this TU is again activated by this GTP molecule with the help of another factor, which is TS. So this TS factor helps in activation of this TU, and so that it may be attached to other transfer RNA molecules for further addition. And what happens next is the most important step, the formation of the peptide bond, and this is brought about by the enzyme which is peptidyl transferase, and you may be surprised to know that this is also a ribozyme. So it means the most important bond in which these proteins are synthesized, the enzyme is peptidyl transferase, and it is a ribozyme. So RNA acting as enzyme rather than some protein acting here for making this peptide bond. And this first amino acid, formylmethionine, is transferred to this second amino acid, and this peptide bond is synthesized between these two. This, and you can see this is the peptide bond, which is C double bond O NH. This peptide bond is C double bond O NH. This is the peptide bond, so which is synthesized here. So this becomes empty. The first transfer RNA becomes empty now, and this is a dipeptide when only one peptide is there, but because two amino acids are there, this is called as dipeptide. And this is dipeptidyl transfer RNA here, and this is deacylated transfer RNA, F-methionine. This in the superscript shows that this transfer RNA contains which anticodon. The superscript is for the anticodon, and if we write name of the amino acid here, that will show which amino acid is attached. So since there is no amino acid is attached, as you can see in this case, F-methionine is attached with this transfer RNA, so this is F-methionine, it's written here, so because it is there. And this F-methionine in the superscript is for UAC. So then translocation takes place. Another molecule of GTP along with this factor, which is EFG factor, elongation factor. And translocation means direction of ribosome movement. So there is a movement of the ribosome that takes place. So this is the direction of the ribosome movement. So when it moves one step towards this side, so this will come at this place and this will come at this place. So this happens here, like this dipeptide, it comes here, and the empty one comes on the exit side, and new aminosyl tRNA that will come on the A side, and this will be the empty one will be released from the exit side, and this process will go on, and this polypeptide chain is being synthesized until there is a stop codon comes here. So any of the three stop codons, if it comes, there is no corresponding tRNA for these stop codons. Instead of that, release factors bind at this place. When this release factor binds to this place, whole thing falls apart. It may be more clear from here that this release factor binds to this place, to this when, because there is no transfer RNA in this case. So when this release factor comes at this place, when with the stop codon, whole things falls apart, and there is another hydrolysis of another GTP molecule. So you can calculate how much energy is required for each peptide bond. And initiating factor 3 that comes in the picture, it the ribosome 2 subunits are detached, and this 30th subunit is ready for another round of translation with maybe with the same messenger RNA, with a different messenger RNA that may takes place. So in this case, this termination of translation takes place in case of prokaryotes. At a given time, there are multiple ribosomes that are attached to the messenger RNA doing this translation process, whether it is eukaryotic cells or those are prokaryotic cells. So here this is an example of eukaryotic cells, 40th and 60th subunits, and this is called as polysome, multiple ribosomes doing translation on one messenger RNA, and there are different polypeptides are being generated. And from where this picture comes, it comes from electron micrograph. It was seen under electron microscope, and from there this cartoon was deduced. So this is the direction of translation with more clarity, and direction of translation, and polypeptides are being synthesized and multiple ribosomes are there. One very important difference between prokaryotic and eukaryotic translation is the spatial difference. Like in case of eukaryotes, both these processes take place at separate places. Like transcription takes place inside the nucleus, and this messenger RNA and other RNAs, they have to come out of the nucleus into the cytosol, or and they go to the ribosome. But in case of prokaryotes, since there is no nucleus, both of these steps or processes, they are coupled with each other. Meaning, this is the DNA, this is RNA polymerase, this is messenger RNA being synthesized. Before this transcription is over, ribosomes are attached to the messenger RNA and start translation. So this is the direction of transcription, and this is same as the direction of translation. So both processes are going simultaneously. So this is called as coupling of transcription and translation in case of bacteria, which is not possible in eukaryotes because of the different compartments. Now, post-translational modifications are very important, and not all proteins are simple polypeptides as they are translated. There are multiple variations that exist in the different proteins. Like some proteins are phosphoproteins, so phosphate group is added to serine, threonine, or tyrosine. Other proteins, they may contain like gamma carboxyglutamate. Similarly, still others, they may contain methyl group like methyl lysine, dimethyl lysine. Similarly, methyl glutamate, trimethyl lysine, and so on. And there are a lot more variations, like some proteins are glycoproteins, other are lipoproteins, some are metalloproteins. So you can find in the textbook what different types of post-translational modifications take place. But I'm not going into detailed mechanisms of all these, but I will go through a little bit of it in the next slides. So this is one example of farnesylated RAS protein. Similarly, okay, another important thing is many of the antibiotics such as tetracycline, chloramphenicol, and this cycloheximide and streptomycin, etc., how they inhibit bacterial growth, or they kill the bacteria. They inhibit translation. If translation is not going properly, those bacteria cannot grow. So these are a few of the antibiotics which are used. Another one is this puromycin, and there is a certain mechanism of each of these antibiotics. Amino-terminal part of the polypeptide usually contains a signal sequence. This signal sequence is present in both in prokaryotic and eukaryotic proteins. If the proteins have to be transported to some other place within the cell, like in eukaryotes in some other organelle, or it has to be transported out of the cell or into the plasma membrane, or in case of bacteria into the periplasmic space. So how these proteins are directed to their destination? This is with the help of this sequence which is present. So you can see this is there are some proteins which contain this signal sequence to the amino-terminal end, and there is a cleavage site, meaning when once this protein is reaches its target, this signal sequence is removed from the protein. Here is one example how it these proteins, eukaryotic proteins, they are directed by the signal sequence into the endoplasmic reticulum, from where they can be exported out further. So this is messenger RNA. Ribosome starts translation, and this is a signal sequence that comes out. Then there is an SRP, which is signal recognition particle, that attaches at this place on the ribosome, and this there is a receptor for SRP on the endoplasmic reticulum and receptor for ribosome also present here. So both of these receptors, they recognize this ribosome and this SRP, and this ribosome along with this protein is plugged in, and protein moves inside the endoplasmic reticulum lumen, and this is brought about due to this signal sequence which is present here. And this blue diamond shows signal peptidase. Once this protein is inside, or it has reached its destination, this signal peptidase removes this signal sequence, and protein is finally released inside the endoplasmic reticulum. This is a dolicol phosphate having varying N number. This is important for post-translational modification in one case, as I will mention here. Here in this case, there is a synthesis of core oligosaccharide of glycoproteins. So in eukaryotic cells, once again, so this is messenger RNA, and as I mentioned in the previous slides, that how this protein is transported inside the endoplasmic reticulum. On the other end, there is a synthesis of carbohydrate synthesis or polysaccharide synthesis, and this is also translocated inside the membrane. Once it is inside the membrane, this dolicol phosphate plays its role, and finally you get this polysaccharide, which is added to the growing polypeptide. So in this way, this is one example. Although I haven't gone through minor details, but this shows a good example how this post-translational modification can take place to make these glycoproteins. And there are proteins which are destined for other organelle or in the plasma membrane or for the secretion out of the cell. Golgi bodies or Golgi complex basically performs this function. So these are ribosome attached to the endoplasmic reticulum, and once this protein is synthesized, they go inside they are exported, and this is a transport vesicle which helps its attachment to the Golgi complex. It has cis side and trans side. So it attaches itself from the cis side, and it detaches from the this trans side, and it is there. From there, it is exported inside the lysosome, or it may go from in the plasma membrane, or it may go outside the cell. So it depends on that. So meaning these Golgi bodies or Golgi complex perform the function of this transport of proteins in eukaryotic cell. This is one example of a nuclear protein, and it has nuclear localization signal. This protein has to be destined to the nucleoplasm, and this protein again, it has some specific factors to which these protein binds, like importance, and they help in the transport of this protein along with these factors, there are certain channels from which these proteins are transported inside the nucleoplasm along with these factors. So once inside the nucleoplasm, these factors are released and protein is destined to its target place. And these factors, they go outside the cell for another round and they are activated over there. So this whole process continuously goes on through this transport channels in the nuclear membrane. So, and this particular pathway, it is activating these proteins or these factors which help in the transport of this nuclear protein inside the nucleoplasm. Here are signal sequences that target different proteins to different locations in bacteria. So as I mentioned that in bacteria, there may be some inner membrane proteins, some periplasmic proteins, or some outer membrane proteins. And these proteins are targeted due to this signal sequence which is present here. And there is a cleavage site similar to the eukaryotic proteins. Once this protein or polypeptide reaches to its destination, this signal sequence is released. This is one model for protein export in bacteria. In this case, this SEC-B recognizes this protein, and there are certain similar sorts of recognition receptors that are present here, as I mentioned for the eukaryotic proteins, and these factors help in the transport of this protein inside this periplasmic space due to the signal sequence which is present here. And you may see that this is also energy dependent process. ATPs are utilized here, and once inside the periplasmic space, this signal sequence will be hydrolyzed. So in this way, this is just an overview of protein export in case of bacteria. Now we have seen how these proteins are synthesized. Their degradation is equally important. Nothing is permanent, even inside the cells. So there is a continuous synthesis and breakdown of the proteins. And the molecule which helps in this is the ubiquitin, which actually targets the proteins which are going to be degraded. So the protein which is going to be degraded, first this ubiquitin is activated with the help of E1. There's an ATP is involved here, which is hydrolyzed to AMP and pyrophosphate, and this E1HS along with this sulfur, it binds to the ubiquitin, and then comes the E2HS, and this E1 is released and E2S is bound here. Now it is the target protein comes here, and target protein is bound to this molecule. So there are repeated cycles lead to attachment of additional ubiquitin molecules here. And this is a proteasome, which is you can say a factory where these proteins are degraded. Once they go inside this, they are degraded. There are many proteases present inside this proteasome. This is the structure of three-dimensional structure of eukaryotic proteasome. And substrate proteins, they come inside, and polyubiquitin, as I mentioned, there are multiple molecules of ubiquitin are attached to these proteins. Attached to the proteins, it interacts with this proteasome, and inside the proteasome, the proteins are degraded. Half-life of proteins it depends basically on on the amino acids present in the amino-terminal residue. So some of the amino acids have more half-life. You can say that these are stabilizing, such as methionine, glycine, alanine, serine, threonine, valine, more than 20-hour half-life. Other amino acids are destabilizing. Some are like 30 minutes, and others, and some like arginine is only it 2 minutes half-life of the proteins where this arginine is present. So it means that protein, once synthesized, as soon as it is synthesized, that is destined ultimately towards its degradation uh, in a certain period of time, and that depends on the amino acids present at the amino terminal residues, and the half-life in case of bacterial or prokaryotic cells is shorter as compared to the eukaryotes. So with this, I finish this lecture on translation. I overviewed this uh, whole of the translation in case of prokaryotes, and also gave some what are the differences with the eukaryotic translation.